Okay, welcome everybody. Welcome to the Friday PowerTalks seminar series. We have here and really proud to have one of our own speaking. And if you have questions or comments, feel free to enter them into the chat. And there will also be a time at the end to further answer questions if you'd like to ask more or speak. So without further ado, I'll present Jake Chen to introduce today's speaker.
All right, well, good morning. Welcome to the seminar series. Today we have Dr. Zongliang Yue. He is currently a postdoc at the UAB Informatics Institute in the AI.Med lab under my supervision. He obtained his Ph.D. from UAB in bioinformatics about a little over a year ago in 2020. And he completed his PhD in 4.5 years, I believe. Prior to that, he was a master student and got his master degree in Indiana University in the School of Informatics. And prior to that, then he also had a number of years of experience as a research assistant, and also got his bachelor degree in China. And he has done tremendous work in developing tools in systems and network biology, and has published, I think, a dozen bioinformatics hardcore algorithm software tools. And today he's going to talk about functional genomics downstream analysis and the use of common bioinformatics tools. Let's welcome.
Thank you for the introduction. So today I'm going to give this topic. And first of all, let me just provide outline. I will briefly just bring up those background of the functional genomic analysis, geneset network and pathway analysis, also known as GNPA, and the 3 generations of the methods used here. So the first question you may be curious about, what is functional genomics? When talking about the functional genomics, it's always talked about some gene functions or protein functions. And once talking about the functions, you cannot ignore the interaction between those genes and proteins. Therefore, this definition comes to this gene function or protein function and interaction in a specific context. So know that there's a phenotype. Usually, it's driven by all different kinds of omic data we processed, such as in the DNA level, we have genomics data, epigenomic data. For the RNA level, we have transcriptomics data. For the protein level, we have a proteomics data. And for the metabolite level, we have a metabolomics data. All this contributes to the phenotypes here. So how we can review this relationship between this genotype to phenotype? Well, very simple, very straightforward model they're using, like for publication early this year in the Cell Journal. They have used the yeast and try to find those isolated strands to do some mathematical model to predict the metabolite fluxes with regard to the phenotype here. The key discovery of this paper, they can do the individual genotypes and predict the metabolic fluxes in the microbiome communities. Specifically in this study, they have bring up this 79 bacterial isolates and sequence phenotypes, and do this metabolite phenotype by also observing this gene presence and absence. And finally, they can do this consumer resource model to predict community metabolite fluxes from the phenotypes. In detail, they have generally produced these 3 steps. in the first step, they measure the phenotype of the isolates, so that they have the culture of the isolate strands, and they do use the denitrifying the isolates and screen for the growth on the succinate defined median under the aerobic conditions. Therefore, they can see those 8-point standard curve, and they can feed this curve with a line here, with a curve here, and see this reduction of these nitrates and nitrite using the consumer resource model. Basically, they can just screen for the biomass density changes and the reduction of the nitrate and nitrite, and therefore they can infer the reduction rates of this nitrate and nitrite. In the step 2, they predict the phenotype from the genotype via the regression. Since they have this different specific strand reduction rates, they want to infer what the genotype will correlate it to this reduction rates. So therefore, they do the genome, the gene screening, and see those gene absence and presence, and do use this the cell regression to infer the coefficient of these genes here, and therefore they can kind of know this hidden latent parameter to do this consumer resource parameter prediction. And in the step 3, they predict the community metabolite dynamic using the phenotypes from the single strands. So with a different combination of the strands here, they can kind of use the inferred this consumer resource parameter to finally predict the curve would be like. So this is a very straightforward models to infer the function using the functional genomics to infer the phenotype would be like from the genotype. But for a complex cases, like a human or mouse model, the functional genomics can review. So there is another paper published early this year in the Cell Stem Cell Journal. They have used a multi-omics analysis to uncover molecular events here. Specifically, they highlight the single cell data they're using. They have this control and the verteprophine treatment. So verteprophine is known as a YAP inhibitor that has the treatment yield wound regeneration in mice. So once you treat with that, it will have not go to a scarring healing, but another trajectory regenerative healing trend here. And in this, they can know this divergent molecular pathway of the scarring versus the regeneration defined by the differential mechanical versus traditional WNT signaling. And with this signaling, they also can identify the key gene driver, which known as the TRPS1 for the wound regeneration in the context of the blocked mechanical signaling. So this is their pipeline to do that for the single cell or this proteomics or the matrix ultrastructure analysis. I will just bring this single cell RNA-seq as an example how the functional genome analysis can help in interpreting those functions. So they have the post-operative days and the treatment of the PBS and the verteprophine. And they do the single cell analysis. They can directly get this a UMAP plot. They also map with different clinical features as cell types they have identified, and also the treatment groups and the individual mouse and the post-operative days here. And what's most interesting cluster they have discovered is the fibroblasts here. So once they have used this fibroblast subcluster, and they can do the monocle 3 information theoretic manifold optimization, pseudo-time branch analysis, and compare the branch correlated genes here. So for the pseudo-time branch analysis, they can infer this trajectory by using monocle 3 and generate the pseudo-time. And they can also discover the sub trajectory that this is red above is the phreatic trajectory, and the down bottom is the regenerative trajectory. And correspondingly, they can discover these genes, correlated genes. For example, I just put the negative correlation with the pseudo-time candidates here. They highlight, for example, this is the TRPS1, the genes they focus on is negative correlated to that pseudo-time, and it's mostly high expressed or overexpressed in the regenerative sub trajectory. And afterwards, they just collect all those candidate genes. They have specified they use the top 1% of the genes negatively correlated with the pseudo-time and do this enrichment analysis. They have using enriched R and put up those gene ontology terms here. So the most interesting one they have found related to the PI3K-AKT signaling pathway, WNT signaling pathway here. And therefore, they just find these driver genes, TRPS1, and do this validation using the experiments like the genes overexpression or the knockdown of the genes, and see the trend with the control. So this is a very typical functional genomics that have evolved in this multi-omics analysis. And instead of using the traditional one, we also have the integrative gene site network and pathway analysis known as GNPA. So for the upstream analysis, as you can see, usually it's like a bulk analysis from the RNA-seq or single cell analysis, or the exome readouts from proteomics or tube-seq data. They do the traditional 4 steps and get the candidate genes or sequencing for a matrices here. But in the integrative omics data analysis, you can see this kind of more complex with such as a clustering to discover the sub-pattern, or network analysis to highlight those critical genes or the events in the network, or the pathway analysis. These have a complex interaction and regulations network there, or the gene set enrichment analysis, which just represent overrepresented gene sets or underexpressed the gene sets there. And this information is already a time-consuming step, and many fragmental usually need a specialized bioinformatization, and all those steps integration is currently immature. So afterwards, once we report some discovery, can do this white lab validation. This is regarded as downstream analysis and publish the data. So the current generation in the existing method are these major 3. Assume there's kind of the input of the pathway database and also the results from like next generation sequencing data. We can do this over-representation analysis known as ORA, functional class scoring FCS, and pathway topology analysis PT to assess the pathway significance. So what in the detail of this? So ORA is statically evaluates the fraction of the genes in a particular pathway from among the size of the genes showing changes in expression. We know that given this knowledge, the 4 numbers here, the genes altogether in the background, the chosen like terms of the gene members in the chosen terms, and we can also use the kind of a hard threshold to get the candidate genes and also get the total number of the hits and the hits inside of the gene ontology term. And therefore, given this format number, we can infer the probability, such as the foreground is a K hits fall into the gene ontology term of the size M. And also the background is the number of the probability choosing the n hits out of the total number of the genes in the whole background. And therefore, we can infer the probability of observed k hits in gene sets. Therefore, we can provide a p-value to estimate how these chosen candidates overexpressed in some overrepresented in some chosen terms. The pros of this technique is you can do any simple sample functional interpretation, and there's no constraints such as you need to always provide any additional attribute. So in this method, it doesn't need. But the cont here is the loss of information. As you can see, these genes are, once they regard as candidates, they are treated as equal. And also no interaction among the genes. There also ignore the correlation structure between the genes, because if we have matrices, there are always some kind of the correlation structure with it that will be loose in this ORA analysis. And each of the pathway is independent of the other pathway. So the most well-known tools for this ORA, like David, enriched R, WebGaussr, and you can just see there's a reference here. The second generation is function class scoring, which different from the ORA, they just get the whole expression matrices with a ranked gene list. Assume they have a different phenotype classes A and B. You can generate the order by seeing the upregulated in a specific class. Therefore, you can run it globally. And therefore, with a selected gene set S, you can see the hit of the gene member in the gene site in the global rank. As well, so for this leading edge subsets, you can see they can perform the sum static based on the signaling to noise ratio, such as like if a gene is not in site S, they will add noise with this penalty. If there's genes inside, they will add the signals here. So all this sum together will accumulated together will be around zero. Exactly 0. So they can see whether it's a height enriched in the in the gene side or the tail reached. So if this is height rate, it will give a positive score here. So the most used methodology here is like gene set enrichment analysis, GSEA, or some other variation like the pathway analysis with down weighting of overlapping genes, PAGE. So the pros of this is no artery threshold. So you can usually observe the weaker but coordinated changes in the same pathway. And also they kind of refine the dependence between the genes. As you can see, if these genes are co-expressed, like overexpressed, they can just reflect in this ranking. And the cons of this is still the pathway is independent of the other pathway. And the typical rank-based statistics using expression or significant can yield some biases, such as if we know some genes have stimulation in the pathway, and some genes may have inhibition in the pathway. So once that's solely consider that as co-expressed, we should think that the genes upregulated in the first category and the genes in the downstream category should have a synergetic effect in the pathway enrichment instead of the agnostic effect there. So therefore, the third generation pathway topology-based approach benefits by this information from the gene interaction, gene regulation. So this is gene sets enrichment analysis regard those genes working isolated, but in the pathway, and always see there's a good annotation of the gene-to-gene relationships. And so this is the methodology have been widely used in the pathway topology-based analysis. And the pros of this is a pathway topology to compute the gene-level statistics. And the cons is model dynamic states. So current annotation is lack of this dynamic. And each pathway is independent of the other pathway still here in this topology-based approach. And also some challenges here is the annotation. Since they highlight the annotation in the pathway topology, but still the low resolution knowledge bases, such as if we know that the GWAS data have provide a lot of useful SNPs, but the SNPs currently we didn't have annotations such as current active genes in the given pathway, whether they can influenced by the SNPs in a specific condition. That information is losing here. And for the incomplete or inaccurate annotation, such as whether the pseudogenes should be considered in the pathway, and that will have an effect in the significant enrichment analysis. And also some of the information getting from inferred from the electronic annotation. So the quality is still a concern here. And the missing condition and the cell-specific information, such as whether this reaction is still existent in the specific defined cell type that doesn't provide in that current doesn't exist in this pathway topology-based approach. So you may wonder what can be improved in this modeling in the functional genomics. So traditionally we have those statistical models such as a hash threading show to just using the Fisher t-test, Fisher test or t-test to report the p-value. And usually the candidate genes is around 100 to 1,000. But with the network, you can always implement with the network structure to increase the coverage and see those upregulated, downregulated genes with locations in the network. But the advanced network will consider those genes not isolated together, but also working in the prior knowledge with the groups. For example, this gene set A, B, C will also have a certain overlap. The genes can highlight the genes in the different functions, and therefore we can generate the genes higher level, like the gene size crosstalk regulations in the advanced network modeling. So GNPA with the pathway annotated gene list and gene signature electronic repository, also known as a PAGER, is what we have contributed to the functional genomics. So PAG is a super gene set which have those integrative information. You can think about each PAG can be a set of the genes plus a curation. The optimal information can be the gene weights, the structure, and data links. So the P stands for the pathway. So pathway level, we can infer from the RNA-seq data, microarray data, proteomic data, and give those structure, the gene network structure. And the annotated genes, that is A here, it's also from this RNA-seq data, but it have a biological curation to say this have some indication in the disease or related to any specific biological conditions. For the gene signature is a relatively low quality, which is a computational inferred result, such as using a cutoff of the p-values to generate the list of the genes. Therefore, we can generate the G-type here. We also have a name convention here. For example, we have this composition of the PAG by using the 3 letters plus the 6 digits here. So with the naming standard, the first letter stands for the type of data, the second position stands for derivation method, and the third position stands for the relation details. Therefore, you can immediately recognize these PAGs by using these three letters here. So FAX stands for the functional genomics with a curated from literature. There's no interaction report in that literature. And we also provide the metadata here to show the detailed information in the PAGER website. What's new in the PAGER version 3? Since we have published 2 older version and what we have improved. So the first is the interactive visualization panel, which provide like a horizontal bar plot to show which the PAGs are highly ranked based on the specific biological conditions. And also we have this scatter plot to show additional dimensions such as quality control. And also we have M-type PAG-to-PAG network, which introduce visualization, interactive visualization using a new JavaScript library. And you can see those color option, like given the source or the type of the PAGs there. So you can play with this once the visualization panel in the PAGER 3. And for the data coverage in the PAGER 3, we also introduce the protein-specific data source, pathway, and tissue-specific expression, interactome, literature curated, and the cell-specific expression here. Therefore, these all new data sets bring up the statistic of this summary. So currently, the PAGER 3 have over the 100 million of PAGs and also 113,000 of PAGs and 42 millions of the PAG-to-PAG relationships here. Another contribution of this PAGER is we have think about whether the PAGs have a high biological relatedness by using the gene-gene interaction there. So we have defined a CoCo score, which called cohesion coefficient. So we just evaluate the significance of this by using the hypergeometric test, which we can count of the background of the interactions. And the theoretical interactions we can infer by also use a total number of the combination, theoretical combination. And for the foreground, we can also infer the theoretical interactions inside of the selected PAG. And we can also use a observation of the interactions in the PAG. Therefore, we can get these 4 numbers to put in the hypergeometric test to evaluate how the significance of this interaction in the foreground, which we define as a co-I. Co-T is apply the same principle, but instead of using the protein-protein interaction, we just consider as a triangles here. So for the background, we can count of the theoretical number of the possibility of these triangles, but also the observed triangles in the background. In the foreground, we can also have the observed triangles with also theoretical number possibility of these triangles forming and put here. So why we introduce a co-I and a co-T? Because we observe that co-I is better for a large size of the PAGs than do the evaluation, but co-T is better for a small size PAGs. So all this co-I and co-T sum together, we can form a CoCo score, which bring up the precision here. And in the second generation of the CoCo score, we have a normalization step here. So before we have see there's some size space as we increase the co-I along with a large size of the PAG. But after the normalization, we can see this have been normalized in the baseline. Same wise for the co-T, we also do this normalization, and therefore we can enable the comparison between a large size of the PAGs, and therefore the CoCo score is increased a lot here for a small size to remove the size bias. Another thing is once we have evaluated the PAGs and well-curated PAGs, can we bring up a whole new level of the relationship to solve the issues like the pathway not work isolatedly. So we have defined the PAG-to-PAG relationship, which called the co-memberships based on the PAG-to-PAG relationship, overlapping co-members there. Simple as we have adopted the same principle using the hypergeometric test, but the four members are different. So the capital N is the total number of the genes in the unions of these PAG-A and PAG-B. And small n is the number of the genes in the set A, the capital K is the number of the genes in the set B, and the small k is the overlap genes there. Therefore, we can do this significant evaluation, and we control the first discard rate using the Bufferoni-Hofburg procedure. And for the regulatory PAG relationship, we define the R-type PAG-to-PAG relationship, which we have a bring in the gene regulation here. We also adopt the same principle, but the number here is the capital N is the total number of the gene regulations. The small n is the number of the genes regulation where the genes in A regulate genes outside the A, which highlights in the yellow color here. And the capital K is the gene regulation where the genes in B are regulated from the gene outside B. And the small k will be the genes regulation directly from the A to the B. Therefore, we can infer the hypergeometric test to infer the p-value, and therefore also we also apply the first discovery for the control. And the extended application of the PAGER, like the PAGER rank, assume we have a list of genes. Can we use this prior knowledge to do this prioritization of the genes? And using this phenotypic network. So in this example, we just get auth genes. There's 680 genetic candidates, and we also get this phenotypic data from PAGER and HAPI database. And therefore, we can do the enrichment analysis, get those highly relevant events, and infer the score of the genes by using the PAG-CoCo score, which is the quality of control of the PAG, and the PAG enriched score, which is a negative log transferred p-value here to infer. And we also consider the genes weights in the individual network in the sub PAG here. And therefore, we can give a PAGER rank result. So once we have introduced a PAGER, what will be the real case study and application of this GNPA with PAGER? The first thing I want to bring up is a bulk data analysis. We have this myeloid-derived suppressor cell MDSC regulatory PAG-to-PAG network to review key immunity impacts. So the input of this is 1,105 differentially expressed genes with 576 overexpressed and 529 underexpressed genes. And therefore, we do the enrichment analysis to get those different color here. So the red color indicates the upregulated scenario and enriched, and the green color indicate the downregulated PAGs we have discovered using the candidate genes. And therefore, you can just see this PAG and gene to PAG and PAG to genes, kind of the relationships reported in PAGER. We can also summarize the upstream of the immunity suppress process, such as activated cell growth and differentiation signal molecules here. And the downstream is related to the immunity suppressed process, such as the process response for cell differentiation, cancer progression, and chemokine signaling, with those highlighted genes CCR5 here. And for the single cell data, the pager can also do additional cell cluster function inference, such as I just put this bone marrow, spleen, blood mixture cells from a mouse identified by the antibody with the hash tags using the 10x sequencing technique. And therefore, we can color this by giving the bone marrow proportion and spleen and blood with a pie chart here in different cluster, cell cluster we identified in this case study. And therefore, we can generally see the trend of this trajectory and see how this cell cluster derived. And therefore, we can use the cell marker to do the enrichment analysis to infer the context here, whether this cluster would be for which function. So for some of these, like this green colored CR6 plus T cells is reported in the blood. This cell marker have been enriched in the cluster 1. Same-wise, we can also pull up this overlapped biomarkers or the candidate genes here to check whether this is a true specific genes observed in the corresponding cluster. So for this subcluster 12, we have seen these 3 genes reported in the CXCR6 plus T cell positive cells. We have seen these genes uniquely expressed and also with a higher fraction of the cells in the group. Same-wise applied to the monocyte basophil cluster here. And with the trajectory, we can also compare those subcluster to another subcluster and see what the significant reported events there. So for example, I just bring up this L12 signaling mediated by the STAT4 highly significant enriched in this comparison and see what the genes drive like upregulated or downregulated in this network. And the extended analysis, we also have considered that PAGER-COV-Run can highlight the critical genes. So we have used the Alzheimer disease network, for example. Some traditional topology-based ranked genes in the network, we can see those highlights and this font corresponding to this ranking. But with GS ranked genes, we see this APOE happening up to the top here. So this is like a beer ranked prioritization of the genes, but with a pager rank, we have the APOE bring up to the top, and if we bring up this top 20 candidates, we can see this rank changes. So lots of the previous ignored genes or the rank lower genes have been bring up to the top here. What will be this top ranked genes function? We have also used a pager enrichment analysis to see the functions. And we got those key terms here. And we also cross-check the BEER database. The BEER database is about entity expansion, ranking, and exploration using the semantic relationships mined from the literature. So based on these predicates, predications consist of the subject, predicate, and object. We can also see the directions and the predicates among those events that reported in the literature and see how this related to the Alzheimer. So some of the terms put up from the pager is highly relevant to the pager the Alzheimer disease by directly mentioned using the semantic relationship, but some are like the drug information are not that directed, but they have in the second layer to see those reaction and how this inhibits cholesterol to just associate with Alzheimer disease. We also use evaluation like information content recall, information content discovery, and literature correlation statistic to evaluate whether the PAGER rank, also here indicate that the GS rank is better than the traditional gene ranking algorithm, and we outperform the others in this in the three measurements as well. So with all those case studies, we also want to enhance the usages by develop a lot of applications here. So one application is a Pager web app here. It's an interactive online application to perform the gene set enrichment analysis and network interpretation of the functional genomics results. And here we provide online data analytics link here. Also, we have a bundle with UCAN TCGA data science, and they have an interactive visualization apps for the biological network analysis. And underlying technique using here is Shiny, PAGER-3 database, UCAN database, and network visualization uses JavaScript libraries. So this is a general workflow in the Pager web app. So you can choose to load our pre-processed data or upload your customized differential express gene table here. And you can select the corresponding column for those information we asked. And therefore, you can generate like a volcano plot and pull up these genes. You can adjust this threshold to filter down these candidate genes. And for the enrichment analysis, we provide a lot of options to seek whether you are interested in the pathway or annotating gene list or gene signatures by selecting the corresponding sources, and also adjust like similarity score, the enriched p-value of the overlaps, and therefore you can generate a horizontal bar plot. And this is supported by the Plotly, so you can do a lot of changes here for the visualization. And as well, we report those detailed informations in the pathway. And also, instead of using the sole pager, we also compare the enriched R and the WebGuster, which are widely used in the enrichment analysis. And we can see this Venn diagram to see which are the consensus pathway, which are the unique pathways reported in individual tools. And we also report this table for the detailed information. So Pager web app offers both global view of the functions and the detailed genes in the gene network. As you can see for our melanoma case study, we have pulled out a melanoma metastasis stage M1C with a nucleoblast RAS visual oncogenes homolog NRAS gene and VRAS murose scarma viral oncogenes homolog B, known as a BRAF gene mutations here. And therefore, we can visualize the PAG to PAG relationship and see what the global interaction between those PAGs level, and also pull out the keywords among this identified cluster using the Loving algorithm. For example, like if you see this is the right corner 3 pathway and what this pathway related to, so it will highlight the keywords here. We have identified those 4 major cluster and report the key discovery here. One of the interesting packs, if you want to zoom in, you can also see the detailed genes and gene network here. So for this PI3K AKT signaling pathway, we observe that this for a response patient, the drug response patient or the non-response patient, what will be the expression scenario here? So you can see for the non-response patients, this collision-related genes pattern is downregulated. As well, we can also find the upstream the regulator is the OSM reported here, and OSM is known to oncostatin M genes in the upstream to stimulate other 5 genes. Since OSM is an interleukin-6 type cytokine to inhibit melanoma proliferation, so the loss of this OSM gene expression can yield the drug resistance and inhibit the activation of the collagen biosynthesis and interleukin-6 family signaling. So therefore, we can just give a more detailed story in this global picture as well. And for another project related to PAGER is a PAGER-COV, which we have a report, the COVID-19 related or COVID related gene sites information. In particular, the PAGs we have collected, there are 11,000 PAGs and around 20 million of the PAG to PAG relationship in the current version. And we have collected 33 data sources, coverage these representing targets, created SARS-CoV-2 gene size, and the immune response gene signatures here. And the details is in the table shown here. We also developed the application like the PAGER-COV-Run, which is a web-based Python script interactive tool for COVID-19 SARS-CoV-2 functional genomics and downstream analysis. Here's a link, and we also bundle this with COVID-19 functional genomic data GO data size. And we also provide a lot of visualization panel for you to get this biological network analysis. For example, this is front interface, user interface of the PAGER-COV-Run. Have the red panel as the operator to see those selection of the tissues. So I just use the peripheral blood mononuclear as an example, and it will show you the condition of the samples, like the moderate, severe, and ICU compared to the control, and what the differentially expressed genes will report in this table. And once we do this PAGER-COV-Run, we can immediately get this pathway, because we only seek the PAG-CAM as a source, also have an additional source. For illustration of the two, we just use the PAG-CAM and see the result and report this highly significant global comparison between the severe case, moderate case, and ICU case with the function of the pathway. And also, if you just choose the detail of the pathway, this here, you can also see the genes with the RP score is important of the genes located in the network, and also description of the genes with the node size here. And you can easily to see what the genes upregulated comparing to what the severe from the severe case to the moderate cases. So one of the pathway we have discovered is a typical NF-kappa B pathway, and they have reported these 2 genes, which you can see that in the ICU case, these genes doesn't have overexpressed, but in the severe and moderate, this SYK have association. So we can infer that SYK, if you have a high expression, you tend to get you can tend to have ICU cases. And another IKBKB is an inhibitor of the nuclear factor kappa B kinase subunit beta, and once you have observed the expression here downregulated, that you will have tend to observe in the ICU cases here. Oh, actually, I observed that this label is wrong. So the first is moderate, the second is severe, the third is ICU. So these two should be shifted. So therefore, you can see the trend here. And another thing is the next generation of the pager would be like. We have think about if we already get those higher level, like a PAG level network, can we provide a multi-level view? Therefore, we have a gene ontology analysis using layer shells for the Go level assignment. And we have kind of detect the different layer of the paths in the PAG to PAG network. I want to show this in the hierarchy structure. Therefore, we can see this PAG enrichment from a general level to a specific level until to a gene level. And therefore, we can map them together using the dimensional deduction technique and map to two-dimensional space. And therefore, we can give a hint of what will be like in a specific level. Also the functional groups here. So for these myeloid-derived suppressor cells, we have these corresponding functional groups, and we can also identify those specific level. So the lower level means it's more specific function level. Therefore, we can identify this highly relevant to the immunological synapses, T cell receptor complex, alpha beta T cells receptor complex, which also validate in the literature retrieval in this. So this is overall of the pager work with those web server, the application, and we also provide demo in the YouTube. And if you're interested, you can also click this link. And I will just stop here, and you can take a screenshot and try out our tools and see whether we can give you more hints using Pager. I'm also welcome to get any question for this talk. Thank you.
All right. Well, thank you, Zongliang. There are questions. We have 5 minutes of time. You can either type your questions in the chat box or just grab a mic. I guess I'll start with questions, and if there's no other. So you've done a lot of work, and do you think that the tool such as Pager is making a lot of difference, or do you think using any tool will more or less give you a similar result?
Well, that's a good question. So we have used a different methodology, such as for the Pager web app, we have compared with the WebGoster and enriched. They have used the same principle using the overrepresentative analysis. And the purely uses technique, our pager can already have a more discovery rate than the reported enriched pathway in the enriched and WebGoster. I didn't show up the result here, the full result here, but we have kind of literature validations and seek whether we can discover more information than the others. But also in the next generation of the pager, we will bring up those in-network weighted gene evaluation and do this functional scoring and with the topological network. I think that will be further interesting to just get a more processed result. And with those specific, like a cell-specific discovered gene-to-gene correlation or gene-to-gene interaction, we can enhance the discovery.
I have one question. First of all, I think it was really great work. How much do you really feel that the hyperbolic is helping you a lot with the gene network? And also I'm working on a grant for Alzheimer's where we're looking at drug repurposing. I'd be really curious to see your Alzheimer's results and do a bit of comparison. We're doing 2 completely different methods, but I think a third method would be really nice to have as well.
Oh, yeah, that's a great question. So, what the hypergeometric test using can help in this, or metabolites can help in the pager analysis? Is that the additional dimension you're asking?
Yeah, so I missed the first part of your talk, so I didn't see that. If there was a slide somewhere where they showed the benefit of the not using Euclidean but using hypergeometric, I'd love to see that.
Right. So hypergeometric test is first generation, and it may have a low resolution in the recognition of the pathway, since it's only report the highly. Sorry. Let me just yeah, go to this. It only discover the hits and without weight. So if there's some more biological conditions with the expression matrices, so it will be helpful to use this functional class scoring. And pathway topology-based approach is based the building brick is functional class scoring, but with the condition of those topology structure. Therefore, if we have additional like annotation of the network under the specific biological conditions, that will be help in precisely extract those pathway and gene set events in this analysis. So yeah, the more data you can use, a more complex and modeling to get the process result, more process result.
Thanks.
You're welcome. And for the Alzheimer's study you have mentioned, yeah, so currently I have used an example using the PAGER-COV-Run, and by using all those enriched PAGs analysis, and we can just kind of prioritize those genes, such as ApoE here. We have ranked at the top, but for other algorithm, or so it depends on the topology itself, cannot give a good indication as we have.
Have you compared those results with something like AlzGene, where they list off some of the currently thought to be most relevant genes in Alzheimer's? Obviously, something like ApoE is at the top of your list, and would be over there too. Or have you made any of those comparisons at all?
For the human curation, we didn't do that, but we have used a kind of discovery matrices to see whether using the whole side of the background and gave the certain cutoff. Yeah, I can show you the details. So you have the whole background of the window size of the genes rank here, and if we use the top certain percent of the genes, do we discover the gene ontology other than the background, and how much have been covered? So that will be like a gene functional relevance, and therefore we can see this content recall, content discovery, which is this yellow portion, which is a specific discovery other than the background. And compared to other algorithms, we have a report to the best performance.
But Zongliang, you also have the PAGER-COV-Run. If you can bring, go to that slide. Can you go to the slides result for PAGER-COV-Run? Yeah, so if you look at the PAGER-COV-Run, the top one is ApoE from the figure.
Yep.
So yeah, I don't know why. So it was unranked, but then after taking into account of all the pathway information, re-ranked based on network, ApoE came up at the top from AlzGene.
Cool. Thanks, guys.
Thank you.
All right, I think we're at time. So let's thank Dr. Yue again.
Thank you for your time.
Thank you. It was great.